it's gone. Okay. Here you go, Mr. Young. These are just the words, not cheating. Yeah, okay. Yeah, hopefully you can read that. Alright, this video is just going to be explaining how a DNA will just replicate and break into two. And yeah, so come on over here, Tate. This is our example DNA. It's just two ropes put together. So basically, it all starts with the helicase. The helicase is just this little thing that hops on, and it breaks the strands apart, just like a zipper on a jacket. So the original point that it started breaking it apart, the helicase, is just called the point of origin. Okay. Another keyword is a replication fork. That's just a fork in the road. You're just driving along, and then it breaks into two. Okay? So the helicase, like a zipper on a jacket, just keeps going down, and it just keeps breaking it apart. Okay? Now, when we get to the end here, I'll show you something. Okay, now, when you have these two strands, one is going to be the leading strand, and then one is going to be the lagging strand. Okay, the difference between those two, it's a bit complicated, so I'll just tell you that later. Okay, so for the new strand that we're going to try and make, it's going to have to go from 5 to 3 end, the 5th to 3 end, because you can't have two strands that go 5, and say this strand is going on the other end, it's 5, and then it goes over here to this end, it's 3. You wouldn't be able to have this one starting at the 5 and going at the 3 also. So for this new one, we're going to have to start at the 5 here, and then 3 over there. So what we would have to do is take a DNA polymerase that would hop on here, and it's just like a little machine that just starts taking all of the nucleotides and start just popping them on, just like a railroad or something. Okay, so the the DNA polymerase 3 would just keep moving it along and just keep following the helicase as it breaks apart. Okay, now that's really simple because it just follows. But now you get to the lagging strand. That's where it gets a little bit harder. The lagging strand, it's not like this one where it goes from 5 to 3 easily, but you still have to go from 5 to 3. Starting 5 to 3. See the problem? You can't start it all the way at this end, because then it would snap and then it wouldn't work. So what you have to do for that one is you would take a RNA primase. Now the primase